So today we will discuss something about the organization of a nerve. So a nerve consists of many nerve fibers here. So these nerve fibers, they are arranged in the form of bundles here, and these each bundle of this nerve fiber is called as a fasciculus. So here in this diagram you can see that these are the many nerve fibers here. These are the many nerve fibers, and these nerve fibers, each of these nerve fiber is covered by a membrane which is called as endoneurium. Each nerve fiber is covered by a membrane called as endoneurium, and a bundle of such nerve fiber is called as a fasciculus. Is called as a This is called as a fasciculus here, and this fasciculus is covered by a membrane which is called as perineurium, and many such fasciculi form a nerve fiber or a nerve. So and this nerve is covered by externally also there is a membrane here. These whole fasciculi are covered by epineurium. They are covered by epineurium. So this is the structure of a nerve fiber. So the whole nerve is covered by tubular sheath which is formed by adular membrane. This sheath is called as the epineurium. Each fasciculus is covered by perineurium and each nerve fiber is covered by endoneurium. Now we will discuss about the internal structure of axon. So axon has a long central core of cytoplasm which is called as the exoplasm, and this exoplasm is covered by the tubular sheath like membrane which is called as exolemma. Exolemma is the continuation of the cell membrane of the nerve cell body. Exoplasm along with exolemma is called the axis cylinder of the nerve fiber. Exoplasm contains many three things. One is it contains a mitochondria, neurofibrils, and exoplasmic vesicles. Mitochondria, it provides energy. Neurofibrils, they are necessary during the regeneration of a nerve fiber when it is injured. And exoplasmic vesicles, they contain neurotransmitters. Because of the absence of nasal bodies in the axon, proteins necessary for the nerve fibers are synthesized in the soma and not in exoplasm, because exoplasm does not contain nasal bodies. After synthesis, the protein molecules are transported from soma to axon by means of axonal flow. Some neurotransmitter substances are also transported by axonal flow from soma to axon. Axis cylinder of the nerve fiber is covered by a membrane which is called as neurilemma. Now, the nerve fiber which is described here till now is a non-myelinated nerve fiber. So here in this diagram you can see this one is the, the diagram A shows a myelinated nerve fiber and B shows the non-myelinated nerve fiber here. So here you can see this particular part is the exoplasm. It is covered by exolemma. This is exoplasm which is covered by exolemma. This is the covering of exoplasm. Now this exoplasm along with exolemma together, they are called as axis cylinder. Now next thing is about the myelinated nerve fiber which is insulated by myelin sheath, is called as myelinated nerve fiber. Now next thing is about the myelin sheath. Myelin sheath is a thick lipoprotein sheath that insulates the myelinated nerve fiber. Myelin sheath is not a continuous sheath, it is absent at regular interval. The area where myelin sheath is absent is called the node of Ranvier, and the segment of the nerve fiber between two nodes is called as internode. Myelin sheath is responsible for white color of nerve fiber. Here in this diagram you can see This one is the myelin sheath. This is the whole myelin sheath here. And the area myelin sheath is regularly absent here. So this area where myelin sheath is absent, this particular area is called as node of Ranvier, where myelin sheath is absent. Or this is called as node of Ranvier. This is node of Ranvier. And the segment where myelin sheath is present, this segment is called as internode. This is called as internode, where myelin sheath is present. Okay. The chemistry of myelin sheath. Myelin sheath is formed by concentric layers of proteins alternating with lipids. The lipids are cholesterol, lecithin, and cerebroside. Formation of myelin sheath is called as myelinogenesis. Formation of myelin sheath around the axon is called the myelinogenesis. It is formed by Schwann cells in neurulima. In the peripheral nerve, the myelinogenesis starts at fourth month of intrauterine life. It is completed only in the second year of after birth. In case of central nervous system, here the neurulima is not found. So here the myelinogenesis occurs with the help of oligodendrites or oligodendrocytes, which are the neuroglial cells. The function of myelin sheath, the most important function of myelin sheath is it is responsible for faster conduction of impulses. Myelin sheath is responsible for faster conduction of impulse through the nerve fibers. In myelinated nerve fibers, the impulse jump from one node to another node. This type of transmission of impulses is called saltatory conduction. And the second function of myelin sheath is its insulating capacity. Myelin sheath has a high insulating capacity. Because of this quality, myelin sheath restricts the nerve impulse within single nerve fiber and prevents the stimulation of neighboring nerve fibers. The next thing is about the neurulima. Neurulima is a thin membrane which surrounds the axis cylinder. It is also called neurulimal sheath or sheath of Schwann. It contains Schwann cells which have flattened and elongated nuclei. Cytoplasm is thin and modified to form the thin sheath of neurulima. In non-myelinated nerve fiber, the neurulima surrounds exolema, and in myelinated nerve fiber, it covers the myelin sheath. At the node of Ranvier, where myelin sheath is absent, neurulima invaginates and runs up to exolema in the form of a finger-like process. The main function of neurulima. In non-myelinated nerve fiber, the neurulima serves as a covering membrane, and in myelinated nerve fiber, it is necessary for the formation of myelin sheath, that is myelinogenesis. Neurulima is absent in central nervous system, so the neuroglial cells called oligodendrocytes or oligodendroglia are responsible for myelinogenesis in central nervous system. So this is about the internal structure of axon. Thank you.